So, yes, very good afternoon students. Here we are to discuss top 1000 questionnaire on botany, aiming for 180 on 180 for NEET 2026. We all know today is the result day of NEET 2025. Most of your students are, I feel like most of you are happy, few of you are dull because your results might not be up to the expected mark. No issues, need not to worry. If becoming a doctor is your ultimate aim than anything, then there is no wrong in taking one more year long term or one more year power step for your medicine. Okay? But one more thing I would like to say to all students is there are so many nice professions as well. Okay? So, if you are aiming to become a doctor, then there is no wrong in taking one more long term. But then if you think that that is the only profession to survive, it is not true. There are many other very nice professions where if you are passionate, you can go for it. Okay? World is wide to give you so many opportunities. So, need not to feel dull or depressed with your results. There are so many opportunities for you. Okay? So, yes, let us get started. This questionnaire session, how did I plan this is, this questionnaire session is planned based on your question strategy from 2019 to 2025, okay, 2019 to 2025, okay, whatever questions are present, from how the questions are framed from different chapters, like statement based questions might be, match the following type of questions, diagram based questions, assertion reason questions, based on the type of questions, level of the difficulty level and the amount of questions that are appeared from each chapter, I have drawn out a pie chart clearly analyzing the papers from 2029 to 2025. Okay? This we have already discussed in our content session as well, where most of the questions are appeared from plant physiology of NCERT level. If we have to consider the botany chapters of NCERT level, most of the questions have appeared from plant physiology. As now, we have three chapters only from plant physiology. Initially, we have to target photosynthesis in higher plants, second is respiration in plants, third is plant growth and development. These three chapters first we have to finish off beta. After this, we have to go for cell biology. Okay? After this, we have to aim for cell biology. From cell biology, 3 units majorly we have to focus: cell the unit of life, biomolecule, cell cycle and cell division. Accordingly, we will go each chapter wise. After that comes our core botany topics. First, anatomy of flowering plants, morphology of flowering plants. These two are very important. After this, then plant kingdom, after that biological classification, then last but not the least, from this chapter living world, hardly one question might appear in every NEET examination. So, this is the complete analysis based on 2019 to 2025 papers from NCERT 11. Next, if we have to see beta from NCERT 12, what are the botany concepts that you get? Firstly, most major of the concept is covered from principles of inheritance and variation and molecular basis of inheritance. Amongst these two chapters, highly questions are covered from molecular basis of inheritance. Around 32% of the questions will come from this, and principles of inheritance and variation, it is 28% approximately. After this, sexual reproduction of flowering plants, around 24% from sexual reproduction of flowering plants questions are asked. Many a times the questions are linked between different chapters, like from cell cycle and cell division, anatomy of the flowering plants will be linked, or cell cycle, cell division, anatomy and sexual reproduction of flowering plants will be linked and asked. Sometimes from plant kingdom, angiosperms, sexual reproduction of flowering plants will be connected and asked. Sometimes you may get connected questions linking up the life cycle of angiosperm and the chapter sexual reproduction in flowering plants. Like that, connecting the topics of different chapters also, there are many questions that are asked. So, based on this analysis, I have crystal clearly I have planned out on how we can exercise the questionnaire, so that we can get the grand success of 180 on 180 in botany for short, gunshot result. How can we aim that? So that for that the clear planner which I can give you is beta, from the chapters, from the flow of level of the questionnaire as we have discussed, first is photosynthesis in flowering plants, next second will be respiration in plants, third will be plant growth and development. This will be 1, 2 and 3 based on the level of questions. So, with this first we will be completing photosynthesis, all about 150 questions, in and around 150 questions we will be covering from this chapter only. After that in and around 240 questions together combiningly, respiration in plants and plant growth and development will be covered. Okay. After this, what we are going to exercise is, we will go for core botany topics. Those are morphology of flowering plants and anatomy of flowering plants, where we are going to exercise both together again 240 questions. Okay. After this, we are going to look into cell the unit of life, cell cycle, cell division and biomolecules. All together of this chapter we are going to exercise total 100 questions. Okay. 100 questions in total. After that, plant kingdom, biological classification, living world. This will be the flow of chapters, and these are the number of questions that we are going to solve from each chapter. Okay. That is why the questionnaire session is very important. Now, you may ask me, ma'am, how we are going to solve the questionnaire session? Just question and answer? Is that we are going to solve? Not at all beta. For each and every question, I will be explaining you the concept in depth. Okay, deep analysis of concept for each question and how you can tackle that question, and if at all they are memory based from morphology of flowering plants, anatomy of flowering plants or plant kingdom, pure memory based questions, I will give you very strong mnemonics where you can remember it just like this. Okay, and you can solve it very easily. Okay. In this way, if we can spare just half an hour each day for botany, rest all time you will be free practicing physics and chemistry. You can give your valid time for physics and chemistry. Okay. If you can finish off biology strategically in very short time, consistently if you are going to give half an hour, 1 hour each day, you can solve that very easily. So, from NCERT 12 if we have to consider, NCERT 12 as I told you, the very first chapter we have to encounter is molecular basis of inheritance, where I have planned and clearly scheduled you 160 questions from this chapter, where I am going to give you completely match the following type, assertion, reason type, statement based type, all these questions I am going to give you beta. Next, principles of inheritance, around 150 questions. In principles of inheritance, majorly I am going to target the numerical based questions as well. Next, sexual reproduction of flowering plants, as I told you, the connecting dots between different chapters. After completion of NCERT 11 and 12, based on the connecting dots between different chapters, only those set of questionnaire also we are going to exercise. And last but not the least, microbes in human welfare, around 80 questions we are going to solve from this chapter as well. Okay. This will be the level or flow of questionnaire practice that we are going to do. Fine. So, as we have discussed beta, the very first chapter that we are going to solve is photosynthesis in higher plants. Okay. So, in this chapter photosynthesis in higher plants, if you consider beginning of the chapter, it begins with early experiments of photosynthesis, right. It begins with early experiments of photosynthesis. Once let us quickly analyze the early experiments of photosynthesis, then it will be easy for you to solve the questions. Early experiments of photosynthesis. To analyze early experiments of photosynthesis with scientists, their discovery and type of experiment, just I will tell you in just one sheet, fine. For this you have to first analyze the standard equation of photosynthesis. As you all know, the standard equation of photosynthesis is what beta? 6CO2 plus 12 H2O in the presence of sunlight and chlorophyll the primary product of photosynthesis. What is the primary product of photosynthesis? Glucose, C6H12O6 and 6H2O, and oxygen is evolved as a byproduct of photosynthesis. This we all know, right? What is the primary product and what is the byproduct of photosynthesis? Do remember beta. Primary product is glucose and byproduct is oxygen. Clear? So now, who said that carbon dioxide is essential for photosynthesis? It is Mole's half-leaf experiment. What is that experiment beta? Mole's half-leaf experiment. So what did Mole's half-leaf experiment say? How did he conduct that experiment? He has taken a potted plant wherein half of the leaf of potted plant which is exposed to sunlight is kept in a beaker containing potassium hydroxide solution. So in this half leaf, whichever carbon dioxide is present is totally absorbed by potassium hydroxide, where KOH along with carbon dioxide it has turned to potassium carbonate, whereas rest half of the leaf is free conducting photosynthesis. When this leaf after some time has taken out for the starch test, it has indicated that half of the leaf which is immersed in the beaker turned to what? Pale color, and half of the leaf to blue green color after the starch test, indicating the presence of starch. So this said that carbon dioxide is essential for photosynthesis, because in the half leaf where carbon dioxide is present, there only glucose formation has taken place. Clear, right beta? Next, second scientist experiment that you have to remember is Jan-Ingenhousz experiment. Jan-Ingenhousz. So what did Jan-Ingenhousz say? Fine. He said that sunlight and chlorophyll, sunlight in the presence of water, sunlight and chlorophyll, photosynthesis happens and oxygen is evolved from photosynthesis. So tell me, which scientist said that oxygen is evolved from photosynthesis? It is Jan-Ingenhousz who said that oxygen is evolved from photosynthesis. How did he conduct the experiment? He has conducted the experiment using hydrilla plant, okay, hydrilla plant, wherein hydrilla placed in a tistle funnel is inverted, okay. Hydrilla placed in a tistle funnel is inverted like this, and this inverted tistle funnel along with hydrilla plant is placed in a beaker containing water, okay, and above this tistle funnel a test tube is placed. Above this tistle funnel a test tube is placed. Two such setups, one under light and one in dark, were placed. So the setup which is placed under sunlight has given out some water bubbles in this test tube, indicating the presence of oxygen. When the test tube is taken and matchstick which is fired is kept over the test tube, it has shown excess burning, indicating the presence of oxygen. So just remember Jan-Ingenhousz experiment using hydrilla plant, and he said that water and sunlight are essential and oxygen is evolved from photosynthesis. Clear? Now next, coming to the products of photosynthesis. Who said that glucose is formed as a primary product of photosynthesis? It is Julius von Sachs. Coming to oxygen, just now we have discussed that oxygen is evolved from photosynthesis is said by Jan-Ingenhousz. But who discovered oxygen? Oxygen is discovered by Joseph Priestley, we all know, in the year 1770, and it is named as oxygen in the year 1774 by Joseph Priestley. What is the experiment name? Bell-jar experiment, right? And third most important point beta, oxygen coming from photosynthesis is said by Jan-Ingenhousz, whether it is from carbon dioxide or water, that is confirmed by C.B. Van Niel. C.B. Van Niel. He is the scientist who said that oxygen is coming from water, not from carbon dioxide. This is being proved, that C.B. Van Niel has conducted bacterial photosynthesis on green and sulfur bacteria. Remember that. Now, I hope you are clear with the key major points from the early experiments of photosynthesis. Fine? Yes. Yes, ma, you can ask your doubt. I don't know who is this. Beyond My Vision is the name. You can ask the doubt which you have. Yeah. Let us exercise question by question. Any doubt or anything you have, you can type in the chat box. I will be clarifying it. So, first question, if you see, the purple and green sulfur bacteria, the hydrogen donor is. Just now we have discussed that oxygen is coming from water, not from carbon dioxide. Oxygen is evolving as a byproduct from water, not from carbon dioxide, is said by which scientist? C.B. Van Neel. Okay, C.B. Van Neel. What photosynthesis he conducted? Bacterial photosynthesis. Bacterial photosynthesis, where in bacterial photosynthesis the electron donor he has used is H2S, from which sulfur is being evolved. That is the reason after conducting bacterial photosynthesis, he also said that from oxygenic photosynthesis which is taking place in higher plants where water is the electron donor, from that oxygen is being evolved as byproduct. Okay. So, C.B. Van Neel conducted bacterial photosynthesis where H2S is the electron donor, from that sulfur is being evaporated. Clear? So, here what will be the electron donor beta? H2S, hydrogen sulfide. Hydrogen sulfide is the electron donor. Next, which of the following experiments showed that carbon dioxide is essential for photosynthesis? You have to answer me. Carbon dioxide is essential for photosynthesis is said by Mole's half-leaf experiment, that is option A. Clear? Priestley's experiment said about oxygen discovery. Julius von Sachs that glucose is primary product of photosynthesis. Fine. And variegated leaf experiment has said that glucose is being formed at the end of photosynthesis. For glucose is formed only in the green parts of the leaf, that is said by what? Variegated leaf experiment. Carbon dioxide is essential for photosynthesis is by Mole's half-leaf experiment. Clear with first two questions, ma? Yes. Third question you look into. In half-leaf experiment, a compound is used which absorbs carbon dioxide. Okay. The compound is, you have discussed right just now, that is what? Potassium hydroxide, where absorbing carbon dioxide it gets converted to what? Potassium carbonate. Fine. And that half leaf where it is completely absorbing carbon dioxide, this turns to what? Pale color after starch test, and the rest half leaf will give blue black color indicating the presence of starch. Okay, this indicates the presence of starch. Clear, ma? Very good. Next, which scientist showed that green substances in plant chlorophyll is located in special bodies called chloroplast within the plant cells. Okay. This is said by Julius von Sachs, okay, because he is the scientist who said glucose is formed at the end of photosynthesis, and he is the scientist who said that chlorophyll, green substances chlorophyll are located in the special bodies called as chloroplast, and in the green parts only glucose is formed. Clear? This is said by Julius von Sachs by variegated leaf experiment. What experiment, ma? Variegated leaf experiment. Clear? Next, which scientist showed that green substances in plant chlorophyll is located? This we have solved it just now. This question you see, Cornelius Van Neel experiment was on green and sulfur bacteria, purple and green sulfur bacteria. Because Cornelius Van Neel has conducted what experiment beta? Bacterial photosynthesis, where the electron donor is H2S. See, from the same concept how different questions are framed. If you are clear with the concept, you can just solve the MCQs just like that. Okay. So, Cornelius Van Neel's experiment was on purple and green photosynthetic bacteria. Okay, this is on purple and green photosynthetic bacteria. Register in your mind that what photosynthesis he has conducted, bacterial photosynthesis, where you can also call as N-oxygenic photosynthesis, and the electron donor is H2S. Take it down right away. Okay. So, he has conducted bacterial photosynthesis, which is also called as N-oxygenic photosynthesis. Fine. So, it is bacterial photosynthesis or N-oxygenic photosynthesis, where hydrogen sulfide is the electron donor. Clear? Next question. So, here you see the following hypothesis: plants restored the air whatever breathing animals and burning candles remove was given by which scientist? That is from Bell-jar experiment. And this statement, well known statement, which is in your NCERT as well, this is given by Joseph Priestley by conducting Bell-jar experiment. Clear, ma? Next, Engelmann's experiment was on. So, here this scientist, that sunlight is essential for photosynthesis is said by Jan Ingenhousz. But which spectra of sunlight is conducting maximum photosynthesis is given by Engelsmann experiment. Let me explain you this experiment precisely, then you will get it easily. Okay, ma'am. So, Engelsmann experiment, first unilateral light which is exposed to the prism. Okay, unilateral light is exposed to the prism, and this, after exposing to the prism, this light is scattered into WebGR. This light is scattered into WebGR. This scattered light is being exposed to green filamentous algae. Okay, this scattered light from the prism is exposed to filamentous green algae, that is Cladophora. Cladophora. Over this filamentous green algae, some aerobic bacteria were placed. Beta. Above this filamentous green algae, what are placed? Some aerobic bacteria are placed. This aerobic bacteria, after some period of time, they got accumulated over blue region and red region of the light, indicating maximum amount of oxygen is being evolved. Maximum amount of oxygen is being evolved from blue and red region of the light, where he said that from blue and red region of the light maximum oxygen is being evolved and maximum amount of photosynthesis is being conducted. Which spectra of light? Blue light and red light. Okay. This is what is said from the E.W. Engelsmann experiment, and from green light the aerobic bacteria accumulation is very minimal, indicating less photosynthesis happened over this spectrum. Now, you can solve this question easily, ma. So, Engelmann's experiment was on what? Green algae, that is Cladophora, which is filamentous green algae. Clear? Next question. Engelmann observed that motile aerobic bacteria accumulated near areas of a filamentous Cladophora which were illuminated by red and blue light. Okay, these are illuminated by what? Red and blue light, but they did not accumulate near a filament illuminated by green light. Over green light, aerobic bacteria did not accumulate because maximum amount of photosynthesis did not happen near green light. This spectra of light is being not absorbed for photosynthesis. From this, he inferred that. What did he convey from this experiment? He conveyed that red and blue regions of light are more effective in photosynthesis. This option is more obvious. He did not say that chlorophyll absorbs green light because there is no oxygen evolution. Oxygen is released from carbon dioxide from light reaction. This is not the point he conveyed. Oxygen released from water. This is also not the point he conveyed. He conveyed that these two spectra of light are being more absorbed, and in red and blue light maximum photosynthesis is happening. Efficiency of photosynthesis is maximum. Okay. Next question, ma. Geeta Shri, if you are having any doubt or anything, you can message in the chat box beta. Feel free to ask the question, ma. Yes. Assertion reason type of question. This is a little application question, ma. Look into it carefully. 6 molecules of carbon dioxide are fixed to form a hexose. Hexose means 6 carbon compound, that is glucose. Okay. So, 6 carbon dioxides are required, 6 Kelvin cycles are required, right? We all know this. So, 6 cycles are required. This is true. In reason, he is saying that 1 molecule of carbon dioxide is fixed to produce 686 kilocalories in photosynthesis. This much of energy is not formed from 1 carbon dioxide. For 1 glucose molecule formation, the amount of energy produced is 686 kilocalories. In assertion, he said very rightly that for 1 glucose formation, how many carbon dioxides need to be fixed? 6 carbon dioxides need to be fixed for 1 glucose formation. This we all know, right? Okay. So, for 1 glucose, 6 carbon dioxides need to be formed. And for 1 glucose, 686 kilocalories is being produced, not for 1 molecule of carbon dioxide. Then for 1 molecule of carbon dioxide, how much energy is produced? That is very simple to calculate. 6 carbon dioxides, it is 686 kilocalories. 1 glucose means 6 CO2. So, 686 kilocalories are being produced for 6 carbon dioxides being running the cycle. For 1 carbon dioxide, what is the amount of energy? Just crisscross it, it is very simple. 686 by 6, which is approximately 114.32 you will get. This much of kilocalories of energy will be produced from 1 carbon dioxide. Okay. So, remember this is very standard statement that 686 kilocalories of energy are being produced for 1 glucose. For 1 glucose, 6 carbon dioxides are required, 6 cycles have to happen, right. So, for 1 carbon dioxide, how much it is? Very simple, 686 by 6 will give you the amount of energy produced from 1 carbon dioxide. So, now you can opt the statement like this is wrong, assertion is true, but reason is false. Assertion is true and reason is false, that is option C. Okay. Let us exercise one more question, last question beta. This question is from site of photosynthesis, and coming up in the next questionnaire session, let us exercise on site of photosynthesis, action spectra, absorption spectra, and specially on chlorophyll pigments as well. Okay. So, now we look into site of photosynthesis. Recognize the figure and find out the correct matching he is saying. So, from chloroplast structure, let me explain you the chloroplast structure in detail, then you will answer it easily. Chloroplast, we all know it is double membrane bound organelle, and inside a chloroplast you have membranous sac-like structures. This membranous sac-like structures are called as what? Thylakoids. These are thylakoids. Thylakoids will be having membrane, okay, that is thylakoid membrane. So, this is the membranous sac-like structure. This membrane is called as what? Thylakoid membrane, or it is also called as thylakoid lamellae. And at the center you have thylakoid lumen. Okay, this is thylakoid lumen and this is thylakoid lamellae. Remember students, in thylakoid lamellae you will be having light harvesting complexes where the light energy is being absorbed in the form of photons. Till here you remember, in next class I will give you more clarity. Okay, so in thylakoid lamellae what is present? Light harvesting complex. This will absorb the light energy in the form of photons. This stalk of membranous sac-like structure is called as what? Grana. One grana is connected to other grana by what? Stroma lamellae. Okay, one grana is connected to other grana by stroma lamellae, and the fluid filled space inside the chloroplast is what? Stroma. Inside this fluid filled space, you have double circular DNA, double stranded circular DNA. As it is having double stranded circular DNA, this is called as semi-autonomous cell organelle. Okay, semi-autonomous or self-duplicating cell organelle. Clear ma? And in stroma you have lipid droplets as well. This is about the structural description of chloroplast organelle. Now you look into the labelings properly. What is this ma? This is stroma lamellae connecting to grana. A is what? Membranous sac-like structures. These are lipid droplets and ribosomes which are present in the stroma. What type of ribosomes are present in the stroma of the chloroplast? 70S ribosomes. Clear, right? Now you look into the options properly. A is what? A is grana, obviously. B will be stroma lamellae. E is ribosomes. And C will be starch granule, true. D is lipid droplet. Option A will be the correct answer. Clear ma? Once look into the question properly, you will also get a clarity. Okay. Everyone got clear analysis till today? So this is about the questionnaire session that we are planning out. This will be a very good tool for you to clear your 2026 NEET exam in more efficient way. Okay. So stay tuned for all these questionnaire sessions beta. These are completely free, accessible for all the students. Okay. And this PPT is available, this PDF will be available in our NEET World app which is in the Play Store. You can download the app, you can access the complete PDF, okay, questionnaire PDF. As such, you will get above 1000 questions for complete botany, including the conceptual clarity. So don't miss this questionnaire series at all, okay. So saying this, that a huge congratulations from my side to all the students who have reached their expectations or who have got a very good result for NEET, and to all the students whose result is not that satisfactory, need not to worry, need not to get depressed at all. World is wide to give you whole lot of opportunities ma, need not to get down at all, okay. So keep learning, improve your knowledge, definitely you will be placed into a very nice position and a very good institute, okay. Thank you.